So I was asked to give kind of an overview, which is going to be really general, about neurotransmitters. And I was given the topic, are these a cause or an effect of bladder sensation? And at the end, of course, we can debate this, but I think at this point we really don't know. So this is my affiliations, nothing to impact on the talk. We know that afferents are responsible for, in fact, triggering sensation, but of course it's more complicated than this. There are a number of sensations in pathology, such as urgency and pain, thought to reflect C-fiber activation, but the types of mediator, the frequency, the amount of mediators that are released, and I'm sure the other speakers will cover this, from nearby cells, which influence sensation, can be altered and augmented in health and disease. And so again, this is a very complicated area. What's unknown really is whether a particular mediator at any time is the cause or the effect. So what mechanisms are really responsible for peripheral coding of sensation? Well, most cells express similar subtypes of receptors, and they can release a number of mediators that are in fact the same. And the cells use multiple pathways, and so this allows the cell flexibility. They can react according to their location and their immediate environment. Again, the cells have flexibility to regulate function, which allows them to respond to complex changes in health and disease. So urothelial cells are a group of epithelial cells that are clearly near these sensory afferents. They can release a huge, vast number of mediators during bladder filling and emptying, which again affects function and sensation. And some of these are ATP, nitric oxide, acetylcholine, cytokines. Again, the list goes on, and we really don't understand where these are released, how much is released, and the subtype of cells that they affect. So I'll just give a few examples. Is ATP, which is known to be a fairly big player, is it actually a cause or an effect of bladder sensation? Well, clearly when ATP is released, one of the largest group of cells that release are the epithelial cells, and it does affect the nearby sensory afferents. Sensitization of these afferents, as many investigators have found in both basic science as well as clinical studies, can lead to chronic discomfort, irritation, pain, and clearly an increase in bladder activity. But ATP also, there's a feedback between the mucosa and the smooth muscle, so it also affects contractile function. And so again, it can set up a precedent where, via local and spinal reflexes, it can clearly affect bladder contractility. In addition, ATP also affects transport. It allows these cells to move substances across membranes. And so in this cartoon, what you're looking at is actually these discoidal vesicles. During bladder filling, this contains membrane, and these vesicles will actually fuse to the apical membrane, increasing the surface area, which plays a role in volume regulation. ATP plays a big role in this. So ATP plays a role in contractile function, sensory function, volume regulation. Nerve growth factor is another example I'll give. Is it a cause or effect of bladder sensation? Well, there's a huge number of studies that have measured NGF levels in urine, have been shown that NGF levels are increased in OAB, in bladder pain syndrome, and a huge other number of pathologies as well, and decreased in responders to various treatments. And of course, if you block NGF using autoantibodies, it prevents urinary frequency. So because of this, NGF has been proposed to be a biomarker and clearly plays a role in sensation. We know that target-derived NGF can alter neural excitability. If you overexpress NGF only in the urothelium, and that's what you're seeing here, you get a sprouting of sensory afferents. And of course, it increases bladder reflexes in these mice. So NGF, is it a cause or an effect of sensation? Again, we don't know. But it's a little more complicated than that because it can sensitize nociceptors by driving these various pain targets to the membrane, such as TRPV1 and sodium channels. It results in an increased sensitivity to various stimuli, so it does impact sensation. But the release of mediators can be affected in another way. Release of mediators can be affected by the physical properties of extracellular matrix proteins. So we know that NGF, at least in the lung and asthma, for example, an excess production of NGF can contribute to the deposition or increased extracellular matrix, increased collagen. And this is, again, a feature in a number of chronic inflammatory diseases, as well as those associated with hypersensitivity. So my lab is interested in how these fibers, these collagen and elastin fibers, change their distribution during bladder stretch. And so you place a bladder using multiphoton imaging, and you can look and see how these changes in recruitment, the types of fibers, and how much they change during bladder stretch. And NGF plays a huge role in this, which impacts sensation. So if you block transmitters with a number of various therapies, such as botulinum toxin, this really underlies the effectiveness of various treatments. And a net result is decreased afferent sensitivity, improvement of sensory symptoms. Again, not one particular transmitter, but a huge number of them, such as ATP, NGF, and many others. So we know that transmitters in general obviously transmit messages between various cell types. The types of mediators, though, of course, between various cells change, and this changes according to the need of the condition. A number of conditions can alter the transmitter levels, from genetic to pathology to stress, which is something we're very interested in. So the gain of the system is amplified. It's amplified in a number of conditions. But all these correlations that we'll talk about today don't really demonstrate cause and effect. So the mechanism of action and the localization of various transmitter substances has provided a lot of information regarding multiple disorders such as overactive bladder, bladder pain syndrome, you name it, and in allowing us to research medications that really what you want is the normal flow of transmitters from cell to cell. So I'll stop here and we'll go on to the next speaker.